We're really happy that Dr. Jonathan Schilling has joined us. Jonathan's an associate professor in plant and microbial ecology. That's in the College of Biological Sciences here at the U. And also beginning very soon, he'll be the director at Itasca Biological Station. And so a lot of us in forestry have a lot of history with that biological station. So I don't know if he's included it, but maybe we'll get to see some red pine or some red pine logs in the slides that Jonathan has. Really one of the key things of Itasca. So with that, I'll turn it over to Jonathan and he can get us started.
All right. Thank you, Matt. I don't know if I have any pictures of red pines, but now I will look for them as I go. Thanks for the introduction. Yep, my name is Jonathan Schilling. The title that I chose for the talk was chosen quite a few months ago, but it was, I don't know. I change up the titles here and there, and so this one is true, but don't get too nervous about the title. But the general talk is with a focus on fungi that decompose wood that are in trees that we would call endophytes. Those are organisms that live in healthy plants. And this term has a lot of different types of organisms that are in there or that would fall under that umbrella. But the ones I'm mostly interested in are those that would cause problems in healthy trees and then ultimately eat them. Oop, page down. Okay. I will quickly tell you a bit about who I am. So I'm a teaching and research faculty here at the University of Minnesota, and I run a lab. This lab is mostly fungus people, so mycologists, and these are people that study in my lab mostly fungi and mostly fungi that are associated with wood as their substrates. And as Matt mentioned, I'm taking on this job as the Itasca Biological Station and Laboratory Director in January. So I'm very excited about that. And I've been here for 12 years. It's kind of a moment where my research has come out of the microcosm a little bit, I think, to be more applicable to large-scale systems and our forests. And so this is kind of a cool moment for me. And it's also a station that I know has this long history with the forestry program here. So I'm really interested in one of my first things in looking at the history of the station, which has been there for 108 years. So I keep hitting the wrong button. So when you think about wood-degrading fungi, you may think about problems in trees, but we also eat a lot of wood-degrading fungi. So if I get questions, you might get questions about sick trees. I get 9 out of 10 questions about what fungi I can eat. So these include shiitakes, maitakes, oyster mushrooms, chicken of the woods, all these things pictured here. So these are all wood-degrading fungi that make a living sustainably using wood as a food source. So these things fascinate me, but they also cause problems in forests. So I've got 3 parts to this. Webinar 1 is just to introduce you to these fungi. And the second part is going to be to introduce you to some tools that allow us to see them when they are hidden inside of a tree. And the third is to give you a case study of some research that we've done to indicate how those tools can be used to track the progress and the success of those hidden lives for the fungi in trees. So the first part is the fungi that you would find in trees. So in Minnesota, the reason that somebody who is in a position like mine studies wood durability, studies fungi, is because that's the dominant decay process here in the temperate and boreal forests. So if you are in the tropics, you'll find all kinds of organisms, including a lot of insects that make wood their food source. Including termites as a massive driver of the overturn of wood. As you go into the temperate forests, the role of insects decreases. We still have things like carpenter ants and carpenter bees here, but you have a dominated process that's dominated mostly by the microbes, the fungi and the bacteria. And as you head into the northern forests of the boreal, it becomes almost exclusively microbial. And particularly any wood or any of this process that's aerobic, which is most of it, it's going to be dominated by these aerobic wood-degrading fungi. So to kind of get into who these organisms are, you can look at some pictures, one of my favorite things to do. So this is a fungus invading a piece of spruce that's been cut and used as a substrate in a microcosm. I think this is a fungus called Postia placenta. But you can see the little hyphae, the little thread-like part of this fungus invading that piece of wood. And those little hyphae are little cells that are like threads that will ramify through the wood cells, traveling mostly down the wood cell lumens. And you can see that they're puncturing through one of the bordered pits in this piece of spruce. But these are fungal hyphae that are largely moving down the lumen. And what they're doing is getting ready to eat the wood. What this means is if you see some of these fungal conchs, if you see a fungus coming out of the tree and you see two that are the same looking kind of conch on a tree, they're connected by all of those hyphae that are growing through that piece of wood, through the tree. So it's one organism. So these are microbes, but they're pretty large. And some of these you could rank in the largest organisms on our planet. So if you zoom in from a standing tree into the wood and look at these individual wood cells, the wood cells are loaded with potential energy in carbohydrate form, and that's what the fungi want, is to get to those food sources. And also yell at me if I'm too slow, too fast, any of the above. This is a cartoon drawing of a wood cell in cross section, and you can see this thing called the cell lumen. Can they see my cursor? Oh, yeah. Okay. No. Okay. There's the cell lumen, and that's the empty space in the middle of the cell. And then there's the thickened gray parts of the wood cell wall called the secondary cell wall. And this is all part of secondary xylem, which is the scientific definition of wood. And these lumen are where you'll see these fungal hyphae growing. And they'll be pressed up against the secondary wall, and they'll start secreting enzymes that are meant to loosen and unlock the carbohydrates that are in wood so that they can eat them. And this is not an easy process. So the blue and green on the slide are the potential components that are carbohydrates that they could make energy from. And then the yellow is lignin. So lignin is there, and it provides some structural integrity to the wood, but it provides a barrier when the fungi want to go in and eat the rest of these carbohydrates. And the rest of those carbohydrates in something like spruce may be 65% by weight of the total mass or the total wood. So there is a lot of carbohydrate energy there that's to be tapped. Yell at me if I'm doing something wrong. So in terms of weight percents, I already said this, but for spruce as the example, you'll get 28% hemicellulose, 42% cellulose. That's all metabolically available energy as long as these fungi can unlock those polymers to make monomer sugars to eat. So it's like candy, but it's locked up in a tough to access matrix of polymers. The lignin is there, and then there is this insolubles or extractives contents, which by weight is only 5%. But if you know about heartwood and particularly things like tropical heartwoods, these are very durable woods that have somewhat low percentages of these extractives, but that are very toxic. So that's why Western red cedar is a durable wood. It's not the lignin, it's the extractives. Here's where I'll introduce these fungi. And the way I'll introduce these is on a piece of spruce. And this is, feel free to fix me. These are three examples that are relevant. So these are all organisms that could eat a piece of spruce at Itasca in northern Minnesota. One is called Phomopsis pinicola, the red-banded polypore. You'll see this on a piece of spruce often on standing trees. This is called a brown rot type fungus. The second one down is Trichaptum aviatinum. This is a white rot fungus that removes components at a similar rate. I'll show you that in a second. And the third one down is called Endophytes tomatosis. All three could be found on the same spruce log in northern Minnesota, but they do this process of unlocking the sugars in very different ways. So if you plot on the bottom the wood mass loss, this is basically over time going left to right, and you look at the fraction remaining of these different components. The way the brown rotters do this at the top, the brown rot, is to remove a lot of hemicellulose early, and then the cellulose at a steady rate, and leave a lot of the lignin behind. And this will leave you with something that looks like this brown, crumbly cubicle rot. So that's a trick where they are extracting the carbohydrates without removing much of the lignin. And this is the focus of most of my biology in the lab. The simultaneous white rot types will remove all of the components at a similar rate, sort of like a progressive erosion. And these interesting selective white rotters will remove the lignin preferentially first. And these have been of great interest in pulp and paper for a long time now. And they actually represent the first of the genomes, the sequenced DNA that we have for the fungi that I study is coming from those kind of oddballs that have been studied for their potential in pulp and paper. So they're lignin selective mechanisms. And there's a range of these mechanisms in the forest. And all of these can be hiding in standing trees, even when they're healthy. So these organisms will sometimes cause damage while the tree is still standing, and the amount of damage that they cause we don't have a good estimate for. But in Europe, in beech forests, that's the only strong citation that I have for that. About 40% of the decay there in trees happens before they hit the ground. And this is a strategy for these fungi to get in and harbor themselves in a living organism until they get a moment to shine and then take over and ultimately eat those carbohydrates. And so if you're familiar with the human microbiome, we've learned in the last decade that we are full of microbes, and we don't know what many of them are doing in there. This would be the case of the tree microbiome, where we have hundreds of isolates sometimes in these standing trees, but we know what some of them do, which is cause pathogenicity or at least saprotrophs that eat dead wood. And the big picture here of the birch, the white birch, this is actually an Alaskan paper birch, but these are two fungi that are coming out of the same tree. One on the upper left is Fomes fomentarius, is the white rot fungus, and the one down low on the right is called Piptoporus betulinus on birch, causes brown rot. Those are two very different decay mechanisms at play in the same standing tree, and they're definitely going to be fighting for territory inside this poor Alaskan paper birch. And when we started noticing this was with a student away. This has been 7 years ago maybe, where we were running some tests trying to pit fungi against each other in the lab. And these were some little baby birches that we cut down from cloquet forest and cut them into rounds. And our controls, we had a lot of treatments involved, but our controls, where we surface sterilized and just let them rot in a jar with dirt on their own, was probably the most interesting. It was definitely the most interesting because we got a lot of decay just in those samples. So whatever was already in there was just as good as the fungi that we were adding back to these systems in decaying the wood. And this is a picture of Zoe holding one of those, and he was really excited about that. So he came into my office and showed me this. This is literally the picture of Zoe when he brought this into the lab, all excited. So we realized that these trees at that point contain these organisms that are capable of degrading wood, and they're getting a head start by dealing with a stressful lifestyle in a living tree to get a head start. Something called priority effects by ecologists. So we like to call this another fantastic title, sort of geeky a little bit for the biologists, but that they carry the seeds of their own destruction, which happens to be a publication from a Welsh group too, so we kind of co-opted that. But it's a neat idea, and it made us think of these questions. How many fungi are already inside these healthy trees? And this was 6-7 years ago, and we had no citations to tell us that. We had research in the Scandinavian countries, and we had a lot of forest pathology research, but all culture-based. So trying to coax out the organisms that were there and see who's there by looking at them once you get them into culture. And we knew from the human microbiome and this kind of stuff that we were probably missing a giant group of organisms that would not grow in culture. So we wanted to know how many of these things are in there, in the healthy trees. How many of those are going to cause problems or at least decay the wood that's there versus other stuff, many of which we don't know what they do. And then what determines their success? Because if you're interested in carbon cycling, it'd be nice to know what predicts that. But if you're interested in keeping trees healthy, we assume that management techniques may be able to utilize this information about what it is that coaxes success versus failure for things like pathogens that are in trees. So this is where I have an artificial break in my part 2 to track these fungi in trees. But if anybody has any questions.
I don't see any questions.
No questions. Yeah. Okay.
Just a quick reminder, if you do have questions, if you type in the chat area and send those to all panelists, we'll get them here in Green Hall and we'll relay them to Jonathan.
And how am I doing for the live audiences? Too robotic or? All right, I got thumbs up. All right, but I'm open for questions. So part 2, basically, how do you see these things? Tracking these fungi in trees. So what I did here was I tried to set this up as something simple, a simple concept for what it is that we're doing by trying to utilize DNA to track these. And I guess I'm old enough to know that there was a shift to molecular techniques that was not always well accepted, in part because there was some overvaluing of the tools when that sometimes I think masked their true potential. And the bottom line with these tools is that they are allowing us to see who's there when we could not before. And it's really valuable. So there are plenty of discussions on the best way to do that and how to best utilize that data. But the bottom line is that it's a tool that's allowed us to see what we could not see before. And so it's opened up a lot of very interesting insights into these processes, including seeing what organisms are already there when these trees are alive, because that's when they are colonizing for a lot of these organisms. So what I've got here is our 4 fungi laid out left to right. This is because I love naming their names, but there's Fomitopsis pinicola, the Trichaptum aviatinum, the Endophytes tomentosus, and then there's one on the right-hand side that is called Fomes fomitarius. It's the tender Polypore. The Iceman had this in his satchel in the mountains of Austria when he got covered in ice after getting shot or with an arrow. Anyway, that's that fungus, the tender Polypore. So these are all familiar from the North Woods. All could be found in the same forest sites. The Fomes is a little bit more of a generalist, but all could be found up north. So these all have, again, DNA. So when you see them as a fungus, you're looking at something that's sort of the equivalent of a flower on a tree. And flowers come out occasionally with some periodicity. The fungi will fruit. All of these really will put most of their fruit efforts into the summer and fall. You might see some of this in the winter, but ultimately you're just looking at part of the fungus because all of the hyphae that are making up the rest of the biomass are inside the tree. And so the DNA allows us to see all of the action inside of the piece of wood. So if you imagine all that DNA down inside of a log, first of all, it's a lot. There's a lot of fungal biomass that's going to be in a rotting log, particularly at mid-stage decay. There's a lot of action in there. And the first utilization of these molecular tools was kind of like fishing. So you would have certain organisms in there that you wanted to know whether you had them there or not. And typically those are pathogens. So if you have a robust sequence that you know, and that being the DNA sequence that you know is from the pathogen, you can go fishing for the pathogen sequence to see if it's there or not. And that would be using a tool called quantitative PCR, polymerase chain reaction. And what this does is it fishes out the DNA. It makes a lot of it so that you can see it on an instrument. And then you get to know if it's there or not. And this is a detection mechanism. And it's a useful tool in plant pathology to go fishing to diagnose problem fungi if you know what you're looking for. If you were interested in all of those fungi. You could have a couple of situations, though, where you might be interested in the whole community rather than going fishing for single isolates. And again, you'll see this in the case study, but you might have, I would say on average have about 150 different taxa of fungi, just the fungi that are in these pieces of wood. And there are probably 1,000 or so bacteria species in these communities. So they're very it's a rich community. So for these 4 fungi, if you had their DNA in equal quantities, then you would have an even playing field, theoretically, of DNA in this system, so that all 4 would be in there at the same quantity. If, however, you had one that was dominating the process, then you may have much, much more DNA from that fungus than these other fungi. And that can make a big difference in terms of what actually happens. So the dominant fungi are going to dominate the process, and that's going to cause the wood to become dominated by the signatures of that organism. And if you're fishing, you may find all 4 organisms in this case, but not really get an understanding of how much is in there, or if they're dominant, and if they're really going to cause problems. And again, if the dominant process is this one on the left, this is the only brown rot fungus in the mix, you'll get brown rotted wood. So if you go fishing for the white rotters, you may find them, but they may not dominate the process. So the tools have gotten more complex, but more community focused. And so this is the protocol for doing something called high-throughput sequencing. And this, instead of fishing for single types of DNA, you fish out the whole thing, and then use the tool to tell you how much of each type is in there. And to do this, I've just put this protocol in here. Number 1 would be to take some part of the tree. This is from a different slide set, but to use to look at what happened in the wood. So you might do pH, or you might do carbohydrate content or lignin content, and then homogenize the other half after snap freezing to do these DNA analyses in step 2. In step 3, you would extract all of the DNA. There are kits for this. I put via CTAB, but that's a little dated too. We now have kits, including one called the Power Soil Kit. It will extract all that DNA out of there. You'll use these things called ITS primers that will specifically amplify fungal DNA, and that will give you pretty well a lot of that fungal DNA. And then you look at your concentration of DNA in number 5, and then step 6 is to send it off to analytical services. So my lab does not have an Lumen sequencer. We just send it off for someone else to do the work with an instrument that I don't know if I've ever even seen. My students probably have, but and that's the high throughput sequencing. And what it'll do is basically barcode all of the DNA in there, and then tell you how much of each barcode you have. And so you'll know the sequence identity as well as the sequence amount in terms of the copies of DNA. And that will give you a sense not only for who's there, but for how much of each. And you can get these tables back. You'll get DNA abundance by the taxa, and then you can start messing around with these to tell you who's there by type and things like that. And so that's high throughput sequencing. And you'll see things in data sets that are an indicator that this is a useful tool that has also become much cheaper. That's probably with quote signs around it, but cheaper than it used to be by a long shot than when I started. And so this is one I took from a Scandinavian group. I can't remember if they're from Sweden. Kubartova, anyway. Using this technique, and you'll see in this some interesting things. Some of these fungi that are not in yellow, there are yeasts that are in these communities. You get odd fungi that normally I wouldn't assume are in wood, or I would never have guessed are in there, but are inside the wood. At least their DNA is being recognized as being there. But the ones in yellow are wood rot fungi. One of them is the brown rot fungus that I mentioned earlier, Fomitopsis pinicola. So you get a sense for how common that fungus is because it's circumpolar. Another one's called Resinicium bicolor. That's a white rot fungus. And then at the top is Heterobasidium parvicorum, and that's a pathogen. And I put that in there because that just showed up in the data sets. They weren't tracking Heterobasidion. They were just looking at DNA taxon. But here in Minnesota, the Heterobasidion irregulare, another anosome root rot, basically our cause of root rot, is number 4 on the prioritized list of terrestrial invasive plant pathogens. So this is one that in these European data sets just kind of shows up, not necessarily driving a lot of the problems there, but it's in the DNA that's being fished out. And then again, here we have this prioritized as an invasive plant pathogen. So if you knew to look for this, then you could go out and look for this. But if you are, like many people, taking these high-throughput sequencing data sets to do all kinds of stuff, you could still look through here, and you might find Heterobasidion with much greater frequency than we would imagine from what we know about Heterobasidion from just finding the mushrooms. So it can be a powerful tool for looking at the whole communities. All right, so this is my second inserted artificial break, if anyone has any questions.
No questions online right now?
I'm boring them to death. Is the total number of people watching diminishing? Okay, that's good.
I guess I kind of have a question about, so with the human microbiome, we know that, or we don't know a lot, but there's some sort of potential health benefits of having a good microbiome.
Yeah.
Is there hypotheses about how fungal and bacterial communities in trees
benefit
have positive outcomes, not just pathogenicity?
Yes. So I'll first say that I'm not deeply familiar with the research, but there are plenty of studies on the role of endophytes, particularly in agricultural crops, in helping them with stuff like desiccation tolerance, but all kinds of stuff in terms of stressors, biotic and abiotic. So I think there's a lot of interest, but there is a lot of challenge in trying to get at which components of those microbiomes are the ones that you might want to target. And that's why things like in the humans, I assume this is a fungal biologist working in trees, but that something like fecal transplants work because you're not trying to be too fancy there. You're just injecting a community that has basically been pre-selected by another human to have the right sets of microbes. And so there is, I think, a lot of potential there in agriculture too. So I would guess yes for the forests. Yeah.
Just wondering, I remember being taught to differentiate key different fungi that were wood rotters of economic consequence, and names may have changed, but like Fungi Can Eat, kind of an upside down horseshoe.
Yeah,
I didn't see that on your list, but maybe names have changed.
The names have changed a lot. Yeah, the part of that due to the DNA sequencing, and part of that I think because the mycologists. It's like a crowd. I don't know, but I think that's the same as the Fomes fomentarius. At least the way you described the way it looks sounds like the Fomes fomentarius, and that is known as the horsehoof, and it's also known as the tender polypore that was supposed to be, I think, useful for maintaining coals for fires and stuff, but I'm not exactly sure. But that's one of the two fungi found on Otzi the Iceman at the Austrian-French border in the Alps. It's kind of a neat story because both that and Piptoporus betulinus were found in a satchel on that person, and they don't exactly know why, but they were very useful for those folks for some reason, but potentially for maintaining fires. But yeah, it's a very common fungus. Piptoporus betulinus very specific to birch, but the Fomes is a little more cosmopolitan, and so you'll find it in places like South Africa on other types of trees. It's more of a generalist, but both on standing trees. It'll be in there on standing trees.
And can you repeat what the question was for online?
Oh, that question was asking if I knew Fomes applanatum as a fungus in a forest tree pathogen or a tree problem. I said yes, but I think it has a different name, and I think it's Fomes fomentarius. Yep. All right. Okay. So here come the case studies. Buckle up, because it's going to get wild. So these are, well, first of all, these are my own research projects. So, of course, I love these stories. But to me, they're pretty interesting because we often assume that if these fungi are in there and that's their colonization mode, is to get in first, then we may not have much control over how this process works if it's all about getting there first, which is a timing thing. And a manager in a forest might hope that there's some environmental variables that would constrict what happens so that you could control the outcome. And so these are two studies that I think are clear indicators that you can control the outcome. It's just that you're dealing with a different set of variables than we have in the past, or at least we're seeing different parts of these communities we haven't seen before. So the first one is, well, both of these experiments are from cloquet. These are for little baby birches that we cut in the primes of their early lives, 4 to 7 centimeter diameter trees. No signs of decay, and you can see pictures of these are the actual stem rounds we took there on the right-hand side. Healthy birch cut and then sectioned into these little rounds that you'll see pictures of here. And then we subjected them in a couple of different studies to different environmental conditions and saw two very different outcomes that suggest environment matters. And this was some research that in the early days of my tenure track days was funded by the Andrew Mellon Foundation. It was a super lifeline for me to do some basic research in an engineering department. And it produced some neat results. So the first one I'll brush over quickly, and that is that we took some of these rounds and just put them right there on the ground at cloquet after bringing some back to look at those DNA-based communities that were inside the wood at time 0 at the beginning. And we left those in contact for, with ground contact for up to 3.5 years at cloquet, and ultimately we got a process there in about, I think it was 20 replicates where every piece of the wood, every piece was all white rot. So everything that happened there was dominated by white rot fungi, and the wood itself showed these signatures of white rot. On the contrary, or complete opposite side, we took some of these stem rounds and brought them back to the lab, and then ran an experiment where we killed all of those endophyte fungi that were in there using sterilization autoclave, or left them. And then we did, like I said, a ton of different treatments that I won't mention because the most interesting results were in the controls. And that was, or is shown here with the gray bars. If we killed everything, we basically got no mass loss of the wood over 5 months of decay. But if we just surface sterilized these rounds and let them go, those endophyte communities that were in the wood after 5 months in a jar with dirt and nothing else inoculated, decayed up to 40% in terms of their mass loss, which is for the world of wood decay pretty solid over 5 months. And so I put together these slides that have a sort of a pictorial representation of what we found when we looked at the DNA. And these are the differences between the left-hand side, which is what it looked like when we first cut it down, and on the right side, which is what it looked like after, in this case, 5 months in a jar on their own to duke it out with no external colonizers. And so this first slide is called species richness, and this is the number of different fungal taxa that were there. And this is, again, this is not looking at bacteria, which would make it even more complicated. So these little dots are all of the operational taxonomic units. These are the taxa in soundwood. So we had on average among these 10 separate trees, these are 10 different trees that were cut to do this, 143 different fungal taxa at the beginning using this high-throughput sequencing. And normally in the forest, what you would see in our forest trial included, you would see an increase in richness over time, where you would have an accumulation of fungi that it wouldn't double, but you would see maybe a 40% increase in the total number of fungi that are in there. But if you just let them duke it out in the microcosms, the number of fungi reduced to 57 on average, meaning two things. One, there's a lot of combat and death in my microcosms. And two, that the wood that sits on the forest floor, when it hits the ground, there's a lot of turnover in the organisms that are in the wood. So there's a lot of death and a lot of colonization that happens out there. So it's, for me, an exciting place to work with the rotters. If you use a tool that we helped develop, but it was mostly Peter Kennedy's lab, and who's here at the University of Minnesota. You can look at the types of these fungi by their functional guild. And so if you look at the ones that are saprotrophic, that could potentially eat the wood, there were 26 there at the beginning, which was about 18% of the number of fungi that were there. And then after decaying in the microcosms, there were 12 left. So there were still saprophytes that were there at about the same percentage. But if you start looking at not just fishing out the DNA as presence and absence, but if you start looking at the amount of DNA for each of these organisms, you can begin to flesh out more information from these data. So this is on the left-hand side showing you abundances by the size of the circle. So the bigger the circle, the more of that taxa that was there. So this is just for all the taxa. You can see a fairly even distribution of the fungal taxa that are in the wood. In terms of their abundances, although there are some that are just barely there, a lot of them just barely there. And after the decay process, you see a few of these fungi that rise to dominance in this system, really dominating the abundance, kind of like the cartoon I showed earlier, the other cartoon that I showed earlier. If you do the fun guild tool to guild these and look at those saprotrophs, then you'll see the saprotrophs also kind of scattered about. But when left to decay, you can see the rise of dominance of saprotrophs. So it's not the rise of dominance of fungi, specific fungi. Those specific fungi are saprotrophs, which not super surprising, but you can look at it, and there it is. So there are a few species that are really dominating that process. If you then look at the rot types of those saprotrophs versus other types of saprotrophs, you can flesh out even more information. So these are the fungi that were there at the beginning. This is their abundances, and the most abundant fungi were white rot type fungi. If you look at who was there at the end, and if you were a betting person, I'm not a betting person, but I would have guessed that white rot would have dominated at the end. And the only brown rot fungus that I know in there that I would ever have seen on Birch would have been that one, Piptoporus betulinus, that was there barely. If I were betting, I would bet that this process would have gone white rot, and that if it didn't, that Piptoporus would have somehow clawed its way to victory and would have been the 44%. But it's not. So we find that brown rot dominates in the system, with one exception of this little white rotter called pneumonia. And so we assume that, if I were to assume that this was going to be a white rot finality, I would have been wrong. If I would have looked at, I'll just go ahead and tell you this, I've got them a little out of sequence. If I were to look at who these are, I'll show you that in a second. Piptoporus betulinus is not there. So it's not in the decayed wood taxa. So it's a process that you would assume based on initial abundances might end up being white rot, but instead it went brown rot. And that's also the exact opposite of what we saw in the field. So the other thing that you can do that is really valuable is you can team these tools or pair these tools to look at the DNA signatures with older techniques that are underutilized in my opinion, but are super valuable. So these are wood signatures in the wood. So here on the left is a piece of birch cut in cross section where part of the wood has been white rotted and part is brown rotted, and you can even see the Piptoporus betulinus popping out of the bottom of the birch. And if you were to look at the wood, there are two easy signatures of brown rot that would be in that wood. And one is the solubility of the wood in an alkaline solution. This is a tool that was developed at the Forest Products Lab in the 50s by a guy named Ernst Cowling. And it's super useful. It was part of his PhD thesis, a huge thesis. And then one is the loss of lignin relative to the overall density loss, which is an indicator if you have a white rot process. If you're brown rot, it's not going to remove much of that lignin. And I've shaded it with a picture here that you can't really see. Kind of messed that up in the slides. But in this wood, basically all of the wood signatures suggested that this was brown rot. So it's connecting these communities here with the outcome in the wood. and determining that it is indeed a brown rot process. And the last couple of slides are to look at these individual fungi. And so this is what was kind of for me like a plot twist in looking at this data, and that's because the Piptoporus was missing in this data set. And so instead, in Birch, where you'll never see Piptoporus, or you'll never see fungi, at least I have never seen another brown rot fungus in there other than Piptoporus betulinus, you see the rise of these other fungi that I knew, but not from doing forest research. And these are two species in the Coniophora genus. I don't know if you know these, Alan. Coniophora pudiana, particularly. It's known in Europe as the Cellar Fungus. It's a really big problem in buildings, in lumber. And then Postea, two Postea species. And so if you study wood decay in lumber and durability in buildings, these are going to be familiar fungi because Coniophora and Postea are very common building degraders. So somehow in our microcosmos, we coaxed this final conclusion out of the system. So what that said to me was that there was some environmental variable here that might look something like the environment in lumber. And so we're kind of at a stage where we're trying to figure out why we would get a consequence like that. But we would never know that without having these DNA techniques to track these communities over time. So the takeaway there is that these organisms that can live as endophytes in these healthy trees have the potential to dominate the process. So if they're pathogens, they may cause problems in your trees. And if you're interested in wood decay processes in general, they may rise to dominance or they may lose out. And we have the tools to track that, but probably more importantly, to reiterate it, we have two experiments using the same baby birch source trees in different environments, and we got a completely different outcome based on an environmental variable, not the fungus or not the fungi. And so the ingredients were the same, but the outcome was different. And to me, what this means is that environment matters. And again, for management, for forests, environment is something that you can control. I would say that without saying like climate, but more like the environment that these fungi see, including how you prune, the age of the tree, the species, and all kinds of things like that. So yeah, environment steers this outcome, and again, offers control. So these are my three parts. The fungi that are there in your trees, these are my artificial insertions. I have no idea if other people do this for their webinar stuff. Part two is tracking those fungi, and then the final part was these two case studies. Again, same source material loaded with endophytic fungi, but different outcomes. And that's what I have. That's what I have. Hope that hits the mark-ish. But yeah, anybody online have questions or?
Not right now, we'll check.
No questions online. Questions from here in Green Hall?
Yeah.
I have kind of a question tied to a comment.
Yeah.
In the long-developed log grading business, that is grading logs once they come through the sawmill, or actually grading the logs as they come into the mill and then before they go through the typical saw.
Yeah.
There are training courses for that, and a lot is provided in terms of guidelines for if you have two of those conchs so close, that section is likely gone versus if they're far apart and so forth. But watching that work, participating in it, they bring up the environment, the local environment variable a lot. And you're cautioned that if you're looking at red oak or black cherry or whatever, before you decide on how you interpret external or end of log characterizations, you need to watch those go through your saw or a sample to get a sense of that local environmental impact. One kind of surface or end defect in one area you go over 50, 75 miles, whatever, to a different watershed or whatever, it can be very different. And so we find the predictability over large areas very difficult. And you can narrow in on it if your area is small enough and the environment is similar. So the environment variable comes up in a very practical sense in terms of evaluation of logs, but it's recognized as being very local. And about the closest they've gotten is it's local, so you better look at that locality with respect to another. And so it's interesting to see your importance of environment being such a factor as well. That's
an interesting comment. So I'll try to reiterate that. So the local variability might be something that you would encounter when grading lumber or when grading logs. In that making some assumptions about how it looks if you move to a different area may be wrong. And I would totally buy that, because there's a lot of discussion, at least in the ecology parts of the decomposers, in local versus regional scale variability. And wood rot is notoriously challenging to predict in terms of the rate of decay. So if you want to model carbon, it's fairly easy-ish to do with plant traits in the leaves and the needles. But when it comes to the wood, if you start trying to guess the rate of decay based on lignin to nitrogen ratios or nitrogen contents and things like that, it gets hard, and the predictions are not reliable, or less reliable. So there's, as you can imagine, a great interest in improving those predictions. And I think most of the focus is beginning to shift on local scale variability, and I think that probably has a lot to do with things that we don't quite understand yet in terms of colonization. And so the tree conditions, ultimately for the endophytes, the trees are kind of the gatekeepers for who is allowed, to get in there and reside as endophytes, and who is trying to get through the gates probably depends a little bit on spore load and the availability for colonizers. And at a place like Itasca is kind of interesting to me if you were to compare that to Cloquet, because it's sitting there facing through one pretty thin strip of hardwoods into the Great Plains. And if you've got prevailing westerlies there, I wonder what kind of spore load a red pine at Itasca would face relative to Cloquet or relative to something in Maine or something. So some of those dynamics are hard to understand, but at least from those carbon modeling efforts, it's definitely seems true that there's a lot of local scale variability. So that's interesting though, because that's like got a human element in there, where if you wanted to do that well, you'd probably want somebody who knew the area, or at least could make those predictions based on some experience there. I've only known one person who did that, West Virginia, but he had done it a long time, and he used to describe it kind of as an art, to doing that. So I'm assuming that's where that definition comes from, is from some deep knowledge of through experience. Yeah. 10 minutes for questions.
Plenty of time for questions. If you have them for Jonathan, feel free to type them into the chat area. I guess I have one, Jonathan.
Yeah.
Kind of related to climate.
Okay.
In terms of, when we think about just rotting wood in general, there's a certain amount of moisture that it might need to rot fully. But then you can imagine, we find logs in the bottom of lakes that we bring and restore and make furniture out of. So with fungal communities, what's that kind of tipping point with precipitation or with moisture? Is it, do you see kind of in terms of species richness or biodiversity of fungi, do you see that up to some point and then that kind of falls off, just as we might think of the wood decomposition?
Yeah, you do. So do you see shifts maybe in the community? So what's the sweet spot for the fungi a little bit in terms of moisture, and do you see variability among the communities based on precipitation gradients? And the sweet spot for an aerobic organism doing business in wood is going to be between the fiber saturation point of the species that it's inhabiting, and when it has too much moisture filling the lumen that an aerobic organism can't get enough oxygen. And so that's kind of your red zone. If you're a kiln dryer, you're trying to dry down through the red zone as quickly as possible without introducing checks and stuff like that. Because you don't want those fungi to get a chance, particularly sap-stained fungi, but to get in there, grow through the lumens, and have available water. So when you're below the fiber saturation point, then the water is not available to those organisms and you will avoid the rot. So that's the sweet spot. But within the sweet spot, you'll have some fungi that are pretty good at cutting it close in the fiber saturation point. That some might be called dry rot fungi. That would operate at pretty low amounts of moisture, and a lot of those are, we know, from lumber degradation. On the flip side, in places like the tropics, you'll find some areas where it's cutting it close on the upper end, and you'll get some what I would call oddballs, but they're actually pretty cool fungi that are a lot of times in the ascomycetes, and they are sometimes soft rot fungi that deal with wet wood. So things like xylaria, which is very common and diverse in the tropics, that we see here as what's called the dead man's fingers, but not too many other species. So yeah, there's definitely a range in who's there in terms of precipitation, and there's a sweet spot. When it's so saturated that it's above the upper end of that red zone or the sweet spot, then you'll move through the soft rotters into the bacteria. So if you pull a log from the bottom of a lake or a big lake and it hasn't had the decay, it'll still have properties that are unique, and those are usually in resonance and how they sound, and also in how much moisture or chemical they'll take up. And that's particularly true in the softwoods because those little bordered pits that separate the cells have pectin-rich membranes, and the bacteria like to eat those. And so anaerobic bacteria will get in and eat those. And it then, when that has happened, it links the cells more fluidly, and so you can move fluids through there easier. And then if you make a set of drums, it sounds better, or this spruce topped guitar, stuff like that. A lot of bacterial degradation processes used to make those instruments a long time ago when they didn't know what was happening. They just knew it was good to sink the wood first. But, yeah, that's a good question. There's another question over here too.
So how do the endophytes survive just in a healthy tree?
Yeah, how do the endophytes survive? I do not know. I just don't know. If you look at a life history strategy, so there's an old thing called GRIME, the GRIME table of life history strategies. It's like your approach to life, if you're a fungus or a bacteria, and some of them deal with stress. Some ruderal, they come in and they just reproduce, they eat all the easy stuff and they move on. And then some are just super combative. And the endophytes, almost all of them are listed as stress-tolerant lifestyles. So that means they're dealing with stress. But beyond that, in terms of how they're dealing with that, over the amounts of time that would be required for a little baby birch to become a fully mature birch and then to die, and then for you that strategy to pay off, it just seems amazingly long. So I honestly don't know. That would I've thought about it before. It would be something that you would need a tool to not only track decay dynamics over time, but you need to track individual fungi over long periods of time. I don't know how you do that. But maybe there's a tool to do that. And you'd have to do that for a long time. So you really have to invest in it. So for forestry people, they probably look at what I'm doing and they're like, oh, that's a quick microbiological tools. You get to do that in 5 months. That's cool. You must, but the microbiology people would look at what I'm doing and they're like, 5 months, that is so slow. What you do is so boring. I guess it's relative, but this stuff takes a long time. So I have no idea. That's a great question. It's a great question.
Other questions online, Emily?
Double check here. No.
All right.
Great. Well, with no other questions, let's thank Jonathan for being part. I think I just saw Madison sent out CFE requests form and evaluation, so be sure to fill those out. And as I mentioned at the start of the webinar, our next webinar is on December 19th, and that's going to focus on urban wood utilization. So thanks for joining us today. We'll sign off now.